Okay, everyone, I'd like to welcome you to the second Retina Australia webinar for 2023, the latest in research from Retina Australia grant recipients. First, I'd like to begin by acknowledging the traditional owners and custodians of the land on which we meet today, the peoples of the Kulin Nation. I also pay my respects to their elders past and present. My name is Sally Turnbull. I'm the Administrative Officer for Retina Australia, and I will be the facilitator of this webinar today. As a participant, your camera and microphone will be switched off throughout. There will be an opportunity for you to ask questions once both our speakers have finished their presentations. If you have any queries during the proceedings, you can type a question to me in the Q&A section. Our first speaker this afternoon is Associate Professor Rick Liu from the Centre for Eye Research here in Melbourne. Associate Professor Liu completed a PhD at the Guei-Sheung Medical University in Taiwan and carried out postdoc training at the O'Brien Institute and Centre for Eye Research Australia, CERA, here in Melbourne. He has extensive experience in designing and developing innovative gene therapy approaches to prevent and delay blindness from major eye diseases, especially for neovascular eye diseases and IRDs, with projects continuously funded by the NH&MRC since 2012. Associate Professor Liu is currently a principal investigator and leads a genetic engineering research unit at CERA. His recent research work has extended into the fields of next-generation gene therapy, focusing on refining these applications to revolutionize ophthalmic care with the latest genetic technologies. This afternoon, Rick is going to speak on RNA-based editing strategies as potential therapeutics for inherited retinal dystrophies. So I'll hand over to Rick. Thank you so much, Rick.
Thanks, Sally. Let me share my slide first. Is everyone can see my slide? So, before I talk about science, first I would like to thank for an invitation to this webinar. This is a great opportunity for us to share our latest research to the Retina Australia community. Also, I'd like to take this opportunity to thank Retina Australia for funding this project. The funding was the really first financial support to this project. It actually made the project from 0 to 1. And in the last 2 years, we've had a lot of progress about this project and making this tool become more viable. And then potentially we can harness this tool for treat various inherited retina disease. So before I start with the science, I'd like to tell you a bit more about why I'm interested in the inherited retina disease and gene therapy, particular for this area. So this is Harry. So a lot of you probably already know him. So he was born with a genetic disorder called Usher syndrome type 1, which is born with impact his hearing and gradually impact his vision. So Harry was also a very first patient I ever met. So when I saw him, I just feel we should do something to Harry because he's still young. So if we can do something for him, so he don't need to live in the dark in his future. So that's why we start to invest our time, invest our energy into the solution for treat inherited retinal disease. So not only Harry, there is many kids suffer with the genetic disease. Those of the genetic disease often caused by genetic mutation or genetic default into a certain gene. So those of gene contribute to various visual function. Since 80, there is about 300 genes has been discovered associated to inherited retinal disease. And what is really exciting for this discovery, and also this discovery also lead to latest breakthrough gene therapy strategy to treat one type of inherited retinal disease called Luxturna, which is being proved by US FDA in 2017 to treat a RP65 associated inherited retinal disease called LCA. So basic in this gene therapy, so scientists harness a non-pathogenic virus called AAV. So AAV are able to carry a normal copy of the RP65 gene, and through the injection into the retina, so infect the retina cell, we call RPE cell, and produce normal RP65 protein to maintain the RPE function and eventually prevent the photoreceptor degeneration in the late. So this is quite exciting. So followed by Luxturna proof concept success into the clinical, there is many gene therapy has been in the clinical trial, use a similar approach. So those approach similar to Luxturna, they also use same AAV virus, carry a gene to different gene to address different mutation in the patient. So that why this is really exciting. So apart from Luxturna, we will have more this type of gene therapy to address different gene for the various inherited retinal disease. However, that's not the case for AAV gene because such as Luxturna or some of gene are quite small, that able to pack into the virus. But some of IRD gene are very huge. And that you can see similar like this, AAV is such a small virus. You won't be able to pack the large gene into the AAV, which means the traditional gene therapy similar to Luxturna approach won't work for some of IRD gene. Indeed, if you look through around 300 gene associated retinal degeneration, so you will find this over 45% of gene are just about beyond the packaging capacity or already big to the AAV packaging capacity. So particular like ASH2A, PCDH15, which is Harry carry the mutation with this gene, or ABC4, which count for the Stargardt disease, which are quite high incident gene to associate to IRD. However, this gene until now, this still cannot be use the same approach like Luxturna. So it means we need a more other approach to address those of the mutation, which is cannot be packed into the single AAV or traditional AAV gene therapy. So writing the genetic codon with the latest gene technology called gene editing technology might be a way to go. So since 2014, our lab been really interested into this technology because we believe this technology got the really powerful potential, which is can offer another opportunity to those to correct a genetic mutation on the patient, and also offer a once-off cure for those of the genetic technology gene suffer from a genetic disorder. So CRISPR-Cas9 gene editing is one of the really latest and also really promising technology. So this technology was initially introduced by Professor Jennifer Doudna and Avenue Charpentier in the 2012. So they actually this technology initially they found from the bacteria is a bacterial immune system. So this immune system is acting as an immune guard to prevent the viral phage infection to a virus. So both repurpose this sort of the system called CRISPR system into a biotechnology. So that allow us to efficient and accurately to edit the human genome. So which is with the high efficiency and even more safe in the past compared to the past technology like talent or other technology. Give a simple explain about this technology. So this technology is comprised of the two components. The first component we call Cas9 enzyme, which is acting as a molecular scissor. So this Cas9 enzyme can be guided by a small piece of RNA we call guide RNA. So that guide RNA can guide this big guide molecular scissor through the human genome to search through the specific area that can complementary to the guide RNA. So and then this Cas9 will act in the scissor to cut the double-strand DNA become a two-piece. So this cutting would then initiate a called DNA repair response. This is where how we repair that. So at the same time, we could introduce a repair template. So, and then tell cell, so the DNA repair you can follow by this template to repair DNA. So we can use this mechanism to actually correct the gene mutation via that template. It looks quite exciting about this technology. Indeed, when this technology come up, this hope was come up with, yes, we can use to cure a genetic disorder. However, this repair system, it's very inefficient in adult cell. It means, so we need a better tool. And subsequently, there's a few of the technology invent based on this CRISPR gene editing technology to have better efficient or more safe, for example, base editing. So the base editing, it means from the world, that is we can use this technology to correct just single base without without introduce a template. So how it does is that fusion a small effector called deaminase to the CRISPR system. So via CRISPR system, they can accurately to search and to find the region that we interest. So that the deaminase can be correct the single base from one nucleotide to another nucleotide, which can contribute more than 50% of the mutation can be corrected by this sort of the technology. And also later on, another technology also come up called prime editing. So the prime editing is similar to base editing, but they use a reverse transcriptase, is another enzyme, and then provide a repair template along with a guide RNA to repair. So both technology is excellently increase the hope and efficacy to address genetic mutation. And so with those of tools, so we could actually now to editing those of the mutant DNA in the genome, in the cell. And then that can be lead to produce the normal second message of called RNA and produce a normal functional protein. That's ideally fundamental. We can cure the genetic disease via this way. But the system never perfect for most of us. So this system was limited by some of the factors, for example, in the eye. So we have to deliver a CRISPR system into the eye to correct the gene in the eye. However, it's very difficult because the system is quite big. So you won't be able to put in a traditional AAV, and you only can put in, like, think about other way. But currently, even the nail particle still not quite well to deliver a gene into the retina. And also because this is correcting the genome. So the system is accurate, but sometimes it will cause some off target. So it might be cause some issue with the editing with the other components. So that might be cause other problem as well. So in this case, so we are seeking for, if we, at the moment, we don't have perfect tool to actually correct genome, can we step down a little bit to correct our messenger RNA, which is more in the middle template product, and that RNA still can produce a functional protein in the cell, so that maintains cell healthy and then prevent the progress of the inherited retinal disease. So the idea of editing RNA is first introduced in 2017 by the Professor Fan Zhang group, so based on the Ball Institute. So in the case, they identified apart from the DNA editor, they also found that in the bacteria, apart from edited DNA, they also have other system, similar system, edit RNA. So they took this tool and then doing the modification similar to DNA editing. So they added a simple the amino to the RNA-based RNA editor form we call RNA-based editor. So this RNA-based editor able to see and identify the area which is we interest and use this code ADA deaminase to change the codon. For example, they can change A to I can become a G, so that to correct the single mutation. So according to the literature, so in most of in overall in the genetics, there's 58% of the mutation out of the mutation is caused by single nucleotide mutation. So this approach that we can address more than half of the mutation into the retinal disease. And another advantage of this system is quite small compared to the traditional CRISPR DNA editor. So that able to allow us to use a virus and long-term deliver into retina. And also RNA-based editor also way efficient than the traditional DNA editor, so and more flexible. So that might be holding a promising to treat retinal disease. So since we received the funding, so we are thinking before we move into a treat real disease, we wanted to see how efficient this system. So in this case, we are this is called CRISPR RNA-based editor that we create in our lab. So at the same time, we also compare to another system called CRISPR-inspired system. So CRISPR systems use bacteria protein to accurately recognize the position to edit. But the CRISPR-inspired systems is like artificial fusion by human component. So this component, because all come from humans, so it could be potentially less immunogenic in general. So, and to demonstrate the editing efficiency, we are use a ray fluorescence as a model to test both systems. So in this case, we had create a ray fluorescence protein, so you can see the fluorescence shining, but we create one of the mutation in the halfway of this sequence. So put G to A, create a stop codon. So it means when the protein translates to halfway, so they stop here. So that won't be any fluorescence coming. So if our design-based editor both of works, so they should be able to create one codon to A to G, so that allow to continue produce the protein in the end. So that would be forming a fluorescence shine again. So in this case, we found that CRISPR RNA-based editing able to correct more than 90% of cell and reverse the fluorescence, which is quite exciting. Unfortunately, the CRISPR-inspired system, which is human combined by human protein, wasn't worked so well. Since we get that sort of the exciting data from the ray fluorescence, so now we thinking that's the right time we move to real disease model. To better prove concept, so we employ a mouse model, so which is allow us to do in vitro validation, and then we can use this as a model to test that the system in vivo. So this mouse we call RD12 mouse. So this mouse carry a mutation called RPE65, similar to the human LCA disease. So, and that RPE65 mutant causes the RPE cell degeneration, and then overall lead to a retina degeneration, So, and that was caused by a single mutation C to G in this case. So we design our base editor to target this RPE65 to reverse that A to G, so that we are able to remove the stop codon and let the codon continue. So that able to create a normal protein in this RD12 mice model. So in vitro, we're doing a really similar work like we're doing in the ray fluorescence. So we test the RNA-based editor along with the search, which is CRISPR-inspired system. So before we go into and then initially we are selecting the right guide RNA. So in the guide RNA design, so we have to have the qualcomm mismatch to the codon that which we wanted to correct. So basically, this is kind of the action tail, this mismatch. So, and you have to make the change in this position. And then so we designed several different guide RNA along with the mismatch. And we found particular that two are pretty efficient. And for the CRISPR RNA-based editor, one is pretty efficient for search. So after that, we are putting this system into the cell engineer with the RP65 mutation. So, and this is pretty complicated, so it might be easy just see here. So for the CRISPR RNA-based editor, we are able to correct a single base mutation in the position that we want up to around 40%. And similar to the result from the red fluorescence, so search wasn't showing quite efficient in this position. And then, so you might curious, is that 40% is enough? So, and then we subsequently look at the protein expression. So we are actually able to reverse the protein expression, even we only reach a 40% correction. So that fits what the idea is, that you don't need to correct every protein, as long as you can produce certain amount of protein that able to, in the cell, that able to drive the function and prevent cell degeneration. So, and then we prove concept in the RP65. Now we are moved to next step to do to address more mutation in the photoreceptor and bipolar. Like in our lab, we are currently working on to address like ABCA4, USH2A, and also the mutation RS1, which is happen in the bipolar cell. So just give you a quick about the idea about the USH2A. So we are creating an USH2A model, so which is similar to human quite high incidence USH2A mutation. So our base editor applied to this model, we are able to see around even higher, around 70%, 65 to 70% correction. However, we do aware once you reach certain higher editing efficiency, you might have some off target along with the area like this. So that means you correct the main on target, so we call on target, but you might have some off target. So to address the off target, so our lab also works on a couple of strategies. For example, we are modify the guide RNA to allow to, apart from the one mismatch, so we also mismatch another off target area with the codon that we don't want. So that the result also showing we are able to minimize the off target here, like compared to the original CRISPR design. So once modify guide RNA, so we can substantially remove or reduce off target in this case. So, and also apart from the modified guide RNA, so we also are working toward another solution called domain-late RNA-based editor, which is we put in, so in the traditional CRISPR gene editor, so we put this called ADA RNA-based editor effector in the both end. But in this case, we wanted to put it in the middle of the CRISPR enzyme. So that enzyme can integrate into the big component, big guys, so they won't move much. So that it would be, theoretically, would be improve the specific targeting window. So, and then we've been trying in several positions to insert that enzyme, and we do found a couple of the position that works pretty well. So, and one of the position actually could substantially remove the off target from the USH2A gene, which is and without impacts on target. So we now are pretty excited about this technology. So we now move into the more validation to the other mutation. Hopefully we can get in a similar result from here. So what is next? So looks like we had a really good tool. However, we would be still a long way to go to make this into the clinical. For example, we need to make engineer more set-based editor. And then since we need to be delivered into retina and as many as retina cells as possible, so we have to improve the delivery efficacy. And also we have to validate the editing efficacy in the retina cell from the patient, which will give us more idea about the how this system works in the real retina cell, and that can be employed by the stem cell technology to achieve that. So this is my last slide. So here I think hopefully I convince you about use this technology as a potential the treatment for IRD. However, so along with the other technology being invented in the past few years, so we think we now had the more choice to address the various genetic mutation that associates to IRD. And then so I think in the end, so I think we are not sell like one particular technology is best, but overall we hope we can develop many this genetic tool, and that for patient they can choose the more suitable technology to address their mutation and then to manage the disease in the clinical. So so that's for today my talk. So before I close my talk, I would like to thank a lot of my lab member contribute this work, particularly in Satesh Kumar. He is a PhD student. He actually did most of this work, and also our collaborator, like from the Department of Optometry or Uni Melbourne, and then from West Australia Eye Institute, and also Menzies Institute, for all your work offer for this work, and also the funding body. And thanks for your attention.
Thanks so much, Rick. It's so exciting to see these new things starting to appear. I will invite our second guest, who is Annie Miller, and she is a PhD candidate from the Lions Eye Institute in Perth. And she's been working in ophthalmic research for over 4 years and has written academic papers and undertaken rigorous research in the field of inherited retinal diseases, working dominantly in the field of cone-mediated vision losses. Her job includes the development of novel treatment options for use in inherited retinal diseases and preclinical testing of these treatments in mice models. Annie is passionate about science communication, and she regularly communicates her research to high school and university students. Annie has a bachelor's degree in pathology and laboratory medicine and a master of clinical pathology from the University of Western Australia. She will complete her PhD in the coming months before commencing her postdoctoral work. This afternoon, Annie will speak on neuroprotective effect of SAHA in retinitis pigmentosa and ask the question, do time and frequency matter? So I'll hand over to you, Annie. Thank you so much.
Thanks so much for the introduction, Sally. So I'll just quickly share my screen. I'll just be 1 second.
Okay, while we're waiting for Annie, maybe we could see if anyone has any questions for Rick. I was going to ask a question, Rick. Just in when you talk about introducing the gene therapy to the eye, and you talk about how it goes into the different cells, I'm assuming that when it is introduced into the eye, it just goes to the appropriate cell, like depending on where the different genes need to go. Is that right?
Yes.
Is that how it works?
Yes. So it's really first we need to identify with what mutation been occurred in the patient. So that can be like RP65 is function actually in the retina pigment epithelial cell. So you have to deliver the component of the therapy to the RP cell. Some of the mutation occurring in the photoreceptor, so you had to deliver into the photoreceptor. So at the moment, so thanks for the viral technology, we are able to harness the AAV vector by introduce different serotype. So that able to target the cell, the retina cell in a different retina cell. So that allow us to target the right cell and to deliver a gene or our CRISPR-based editor into the right cell to correct the mutation or to supplement gene in the right cell. So that would be maximum the therapeutic efficacy to right cell. and also that's also a big challenge for the talking about the DNA editing. So for DNA editing, because it's quite big, so we couldn't use a virus as a vehicle to deliver it. So we only can use most of this day is use particle, called lipid particle or nanoparticle. Those of particle might not well addressed to deliver in the right cell, right position. So that's how it makes difficult to apply this technology to the eye. But for the other organ might be easier than the eye. So that's why we are thinking move to the RNA editor than the DNA editor, which is small. You can put in a virus and it can deliver where you need to.
Yes. Yeah. Okay. Fantastic. Okay. I see Annie back.
Yes.
Let's see how we're going, Annie.
Alrighty. So thank you for my introduction before, Sally, and thank you for obviously Retina Australia for providing us with the funds to undertake this research over the past year and a half or so. So today I'll be presenting a talk about the neuroprotective effects of SAHA in a couple of models of retinitis pigmentosa. So just briefly, so our lab focuses on the study of inherited retinal disorders, and we especially focus on cone-mediated vision loss. So when it comes to treating inherited retinal disorders, there's lots of, there's actually not very many treatments available to patients, but there's lots of different treatments available, or sorry, not available, happening in vivo in the lab at the moment. So if you look at two broad categories, we have gene-dependent therapies. So those are things that might involve gene replacement of a faulty gene. So that's things like AAV-based gene replacement therapy, like Lux-Turner that Rick mentioned before in his presentation, which is the only FDA-approved therapy for people with inherited retinal disorders. We also have gene-independent therapies, which are being developed at the moment, mainly in a lab basis. So there's lots of kind of different areas to that. We might have things like retinal prosthesis. We might have things like cell therapies, replacement of cells. And we also have pharmaceuticals, which might be there to, say, prevent the cell loss that happens in these diseases. So we focus a little bit on these gene-independent therapies, and we mainly look at neuroprotection, and we work in a group of drugs called HDAC inhibitors. So I'll go a little bit more into HDAC inhibitors in just a moment, but we'll really briefly just talk about retinitis pigmentosa itself. So when we have RP in patients, the usual initial symptom is a night vision loss, and then there's a peripheral vision loss associated afterwards. So some people don't progress towards total vision loss, but it does vary depending on the patient. It's associated with mutations in over 90 different genes. So it's very genetically heterogeneous, lots of different causes. And usually we have a primary rod photoreceptor cell death, which is the kind of cause of that initial night vision loss. And then eventually we have the cone photoreceptor loss as well, which follows, which results in the peripheral vision loss and the total vision loss in some people. The unfortunate part about retinitis pigmentosa is that once the photoreceptor cells have degenerated, they can't regenerate anymore. So once they're gone, they are gone forever in that patient, and they will lose their vision. So lots of different studies have looked at the causes of cell death in RP, because that's also not very well understood. When it comes to looking at these causes of cell death, there's lots of different avenues that you can look at. We have things like apoptosis, which is programmed cell death, necrosis, which is when a cell is kind of falling apart, not doing its job very well, so it just dies because the body goes, okay, that's not viable. And then we also have autophagy. So these kind of three different main areas of cell death are kind of all considered to have a part in RP, and potentially they all kind of act together. And this kind of cell death might sit in a bit of a spectrum between these three different areas. So there's still lots of work ongoing in this field, but about 10 years ago now, this study actually looked at 10 different models of inherited retinal disorders. So some retinitis pigmentosa, some achromatopsia, and then a Leber's congenital amaurosis model. And they identified that there's quite a few of these non-apoptotic cell death markers that have been upregulated across all of these models. So here we have just a little bit of like a schematic showing some of the things that are upregulated. When we have a mutation, it can cause this accumulation of this cGMP molecule. And then this has other effects in the cell that could eventually cause the cell to die. So we have, say, for example, if we go down this route, we have an increase in our PKG molecule, this increase in our HDAC or histone deacetylase molecule. Eventually it goes on to this PARP molecule, and then we have death. So it is kind of like a cascade, and all of these might have involvement in that ultimate death. So what we've done in our lab is we've focused specifically on HDACs to see if we can modulate that, if we can reduce that increased activity. Is there a way that this could potentially, cause a protection of the photoreceptor cells in our models? So here we have just a schematic showing what actually happens with this HDAC molecule and what's its purpose inside of the cell. So we have here a diagram showing our DNA, and this DNA is wrapped around these histone proteins. So the histone proteins basically allow your DNA to wrap either tighter or looser, depending on what the cell needs. And we have our HDACs and our HATs or our hats, which modulate whether it is tightly coiled or loosely coiled. So our HDACs will make, the DNA more tightly coiled. This results in transcriptional repression, while a histone acetyltransferase, our hat, will result in a more relaxed chromatin. The idea of having a HDAC inhibitor come in, this type of drug, is to actually block the HDAC. So you're allowing for a more relaxed chromatin and more transcriptional activation. So what we look at is pretty much looking at these HDAC inhibitors and seeing if these are going to be a viable gene-independent therapy for IRDs. Now, the particular HDAC inhibitor that I'm going to be talking about today is called SAHA, and it's an already FDA-approved drug that's been used to treat cutaneous T-cell lymphomas, and it's been mainly used in actually cancer research for many years. But there's a few studies recently that have shown that it might actually have a beneficial effect in neurodegenerative disorders, as well as retinitis pigmentosa and other inherited retinal disorders. And so far, they have looked at a model of RD10, a retinitis pigmentosa model, and they have looked at the effect, and it does appear to be neuroprotective in that particular model. So as we can see on the right here, we have on the left our untreated RD10 retinal explants. And then if we treat with SAHA, we actually have an increase in the number of photoreceptor rows. So this is mainly the rod photoreceptors being protected. Okay, so because of this kind of these previous studies and the fact that HDAC overactivity seems to be consistent over quite a few different models of inherited retinal disorders, we now ask the question, can SAHA improve vision in our RD1 mouse model in vivo? So our RD1 mouse is a mouse model of autosomal recessive RP, and it has a mutation in the PDE6B gene. So this gene is just involved in rod phototransduction. In this model, it's actually a very, very fast degenerative rate. So both rods and cones are pretty much lost by the age of about P24. So we see our rod loss starting at about P8, and this is postnatal days, so very, very early in life. And then your cone loss will begin around P14. We've just shown a picture here, just showing what a wild type or a healthy adenonuclear layer should look like at 24 days of age in mice. And you can see it's quite thick here. It has these cone photoreceptors sitting all through here. And then if we look at our RD1 over here, you can see how degenerated it is. You can't even discern those cone cells anymore. And there's almost no rods left at all. So to help answer our question, this is the treatment regime that we did for our mice. So what we did was we decided to do a P12 injection. So we wanted to inject our mice a little bit before that cone loss has actually started to see if we can preserve those cone numbers. So we performed an intravitreal injection, and we either did that with a treatment or we did it with a sham injection as a control. We then waited 4 days and then we collected optomotory testing data. I'll explain that just a little bit more in the next slide. And then we also collected for a couple of other different experiments. We also did some longer term studies where we injected at P12, same thing again, either sham injection or SAHA injection. And then we also then collected optomotory and other experiment data at P24 instead. So that's 12 days after we've administered our treatment. Okay, so for a little bit of background about our optomotor testing, this is a really cool technique that we have where basically we can put mice into like a box and it has 4 screens around the mouse. And what it does is it kind of assesses their visual acuity, because obviously with mice, we can't just ask them to read the letters on a chart. We actually have to assess it in more of an objective manner. So these screens will have these lines that you can see here, and they'll move around and we'll assess if the mouse is tracking the lines as the lines are moving. I have a little video here which would hopefully demonstrate that for you. You see the mouse on the platform, and then we have some lines appearing on the screen. And then if the mouse turns its head, it will indicate it's tracking and it can see the stimulus. So what we saw in our RD1 mice is in scotopic readings, which are your dark adapted readings, which assess your rod function, there was almost no response, because these mice have lost almost all of their rod photoreceptors by this age. So this was not very surprising. And you can see that our wild type is drastically different compared to every single one of our RD1 groups. We also looked at the photopic optomotory data, and these mice at this age do retain a little bit of their photopic or their light adapted visual response, although it is still really low compared to wild type. So here we see that uninjected and sham injected optomotory testings was a little bit reduced compared to our treated, especially this 1 micromolar SAHA here. However, this result was not statistically significant. So we did notice as well, though, that a lot of our mice actually appeared to have absolutely no response to the stimulus that is coming around. It might be because potentially their photoreceptors have generated too much at that point. But if we remove all of the ones that sit on this 0, we can have a look over to the right here, and we might have some potential non-responders excluded from our group. And we can see that this trend is looking a little bit more likely now, although we still don't see any statistical significance showing that it definitely is increased compared to our controls. So we also assessed histology and looked at some staining of the retinas after they've been treated. So when we do our histological assessment, what we do is we look at 4 to 6 different locations in the retina, and we have a nice picture here of a retinal section, and we have the 4 different locations that we kind of locate through finding the optic nerve. So we have our central superior here, our peripheral superior here, central inferior and peripheral inferior. So that's what these different labels here mean. Now, after we injected, we collected 4 days after, and we found that most especially in the central retina, there was an improved outer nuclear layer thickness after treatment. We noticed especially that this increase was mainly seen at a lower dose of SAHA, because we assessed 2 different doses, both 1 micromolar and 10 micromolar. So at this point, it looks a little bit more like our lower dose is actually beneficial, while our higher dose might not be. We also looked at this other marker here called GFAP. And GFAP is an active Molliglia marker, and this usually indicates a little bit of retinal stress in the retina. So in these types of mouse models, you'll usually see quite a lot of this GFAP because there's a lot going on in the retina, a lot of cell death and degeneration. So we see that after we treat our mice with 1 micromolar SAHA, there's actually a decrease in this GFAP staining in our retinas, but we don't see that effect as much when we look at our higher dose, our 10 micromolar SAHA. So when we have a look at these results 12 days after treatment, we see that we've lost this potential treatment effect. So our scotopic and our photopic optomotory data is all at 0. So there's no response at all. They're completely blind by this point. And then when we assess our outer nuclear layer thickness as well, we also see no biologically relevant changes at this age. So it looks like the treatment effect is lost. So this data looked really, really promising, but it's quite hard to work in a model that degenerates so quickly because you have to be very precise with your collections. And, you know, if we might have missed the treatment window, the ideal treatment window by a day or two, then we're not going to see much of an effect. So what we wanted to do is we wanted to look at a less severe model as well as our quite severe model. Here we have our rhodopsin P23H mouse. And this has a mutation in the rhodopsin gene, which is also involved in our rod phototransduction. This is an autosomal dominant RP model. And what happens is this aberrant rhodopsin protein called P23H accumulates in the cell and causes a reduced level of phototransduction. We end up with a much slower rate of degeneration. By around P35, so 35 days of age in these mice, the rod outer segments, which is like the top portion of the rod cell, they begin to shorten. And then by around 2 months of age, P63, we do see that there is fewer rod nuclei found. So here we ask, can SAHA potentially improve vision in this less severe rhodopsin mouse model? So because we've never worked with this particular model in our lab, we only just got it in last year, we wanted to do a little bit of validation ourselves to check that what we've read online in different papers is also what we're seeing in our mouse model. And we see here this timeline of degeneration. We have 1 month and 2 months, which look quite similar to each other. And then we start to see around this 3-month mark that our outer nuclear layer, which is our kind of blue, the blue staining, is starting to decrease in size. 4 months and 6 months do look quite similar as well to 3 months. And we can see here in our figure that there is actually a significant reduction in the outer nuclear layer thickness between 2 and 3 months. We also looked at electroretinogram responses. So this is the ERG response, and this basically detects the cell's response to a light stimulus. We have our scotopic A-wave responses, so our dark adapted response, which looks at the rod photoreceptors mainly. And we see that around this P60 mark, so that's 2 months of age, there is a significant drop in our in the mouse's response to this light stimulus. We also see a similar thing for our scotopic B-wave at P60 at 2 months. And our scotopic B-wave is a little bit more involved in some of the other cells in the retina, such as the bipolar, amacrine, and Muraglia cells. We also had a look at the photopic A-wave, so that's our cone photoreceptors and their response. And we see that this kind of drop in function is a lot later, and it looks to be more around this 4-month mark here. Same thing as well with our photopic B-wave. So what we did next is, because we identified that there seems to be quite a decrease in the outer nuclear layer thickness at 2 months of age, we wanted to administer our treatment at this age to see if we could protect this, this loss. So at this point, we're looking a little bit more at rods because by this age, the cones are not as affected as the rods. And we see that if we apply our drug at 2 months of age, we collect 4 days after, we can actually see an increase in the outer nuclear layer thickness, as we can see in the images just here. And we can also see with just the diagram on the right. So currently, this is our preliminary work. We've only done a small number of biological replicates, but we are increasing them at the moment, and we're seeing a really lovely trend towards the fact that SAHA may be beneficial in this model. So just to summarize what I've talked about, so we are looking at this drug SAHA, and we're thinking that it actually does have a very good potential neuroprotective effect in both of our models of RP. So we noted a significant retention in the outer nuclear layer thickness in both of these models after being treated with the drug. So treatment with SAHA in the RD1 model also appears to reduce the retinal stress marker GFAP, but we don't unfortunately see much of an effect when we look at 12 days after treatment. So they seem to be lost by this point. Looking at ongoing work for this, because it's still a little bit of work in process, especially the rhodopsin line data. What we want to do is we want to assess the cone numbers after treatment as well, especially in the rhodopsin line. So obviously in our RD1 mouse, it can be quite hard to do that because you could see in the slide before just how degenerated that retina is. But we also wanted to assess if multiple injections of SAHA will increase that window of time that we can prevent this loss of cells. We also are looking at assessing any neuroprotective effects of our drug in other models of IRD, because this overactivity of HDAC seems to be prevalent in quite a few different models of IRD. And especially I'm looking at some achromatopsia models for that purpose. So just a quick thank you to everybody in my lab who's contributed to this work and my co-investigators on this grant. So Dr. Olivia Cavallo and Dr. Rabab Rashwan. And thank you all for listening, and thank you all for being patient during my technical problems at the start. Thank you.
Thank you so much, Annie, for that. And I'm glad we finally got it to work in the end. I want to thank both our speakers, both Rick and Annie, and thank you to all of you for coming along. We've got a little bit of time left just at the end now for some questions. If you'd like to ask a question, you can type it into the Q&A section at the bottom of your screen, or you can use the raise hand option at the bottom of your screen, and we will respond to those in the order in which they appear. When we say your name, we'll enable your microphone and you'll be able to ask your question. You'll just need to unmute yourself at your end. And then after your question has been answered, we'll turn your microphone off again. To raise your hand, you can select the option at the bottom of your screen, or you can use Alt-Y on the keyboard. If you're on the telephone, you can press *9. And if you're on the telephone, to unmute your microphone, you need to press *6. And you can also use the unmute button on your screen or just press the space bar. Alternatively, just put a question into the Q&A section. So if people have questions, we'd love to hear from you. And while we're waiting, I'll ask a question. So Annie, I just wanted to get a sense, is the idea just that because these effects run out, that you'd have to, in real life, this would be an ongoing treatment. This would be something that you'd have to keep treating to keep the levels of the drug at a certain level or something so that you would have an effect. Is that how that would work?
Yeah, so definitely. We see this a lot when it comes to, say, treating macular degeneration. So a lot of people have to go into the clinic about once a month to get a top up of the drug that they receive by intravitreal injection, because the drug doesn't last forever once you put it into the eye. Other studies do look at taking things via, say, like a tablet, which would mean that you don't have to go into the clinic all the time, but it's less effective in that form. So for that kind of purpose, we do look at multiple injections, and also looking at some other technology that might, be able to provide the drug on a continuous basis, such as some nanoparticle technologies, which are coming up in the field at the moment.
And when you say nanoparticle technologies, how would they be administered?
So they get administered similarly. They can either be done via an intravitreal injection or a subretinal injection, which is just as a more complex surgery, that subretinal one. But then once it's been injected, it can then continuously supply it to the retina, hopefully. Okay, great. I had another question. You were talking about the different breeds of mice that have these conditions. I'm assuming that there are a whole lot of other breeds of mice that have been bred to express the genes for achromatopsia or other things. Is that correct? Yes, that's correct. So we have quite a few mouse models in our lab at the moment that are achromatopsia lines. And that's actually where I predominantly work, and we test a lot of these drugs in those lines too. Fantastic. Okay, I'm not sure if does anyone else have a question? I can keep going, but I feel like it should be, other people should have a go. Rick, have you got a question?
Yes.
Yeah, I got a question. Just extending from your first question. So it looks like if for hashtag inhibit, you have to give a continuing treatment almost for whole life. So we know that a lot inherited retina disease, retina start to degenerate since they're very young. So hashtag inhibit actually can be inhibit a lot the gene which is contribute to the development. So how you can make the balance between not to suppress about the gene contribute to the development while you protect the retina degeneration at the same time for the very young kids?
So that's definitely a good question. We haven't exactly considered all those things because you're right, it's a very complex drug to provide to somebody because it does lots of different things to the cell. There's different expression at different ages, but it's definitely something that we would consider in the future if we were to bring this towards clinical trials or potentially look at it in our mouse models before we do that. Thank you.
Thanks.
I've got a couple of questions. Rick has asked, is SAHA an abbreviation and what's in it?
So SAHA is an abbreviation. I don't know the full name because it's a very scientific name. And it's also a very complex chemical, so I can't exactly tell you exactly what's in it. But yeah, it's a HDAC inhibitor, so that's basically the kind of purpose or the function of the drug.
And another question from Julia Hall. She wants to know, is HDAC an expensive treatment, for example, in its current use in other diseases? And what would be the dosage comparisons that they use and that you tested for efficacy?
I also haven't checked the price of that, but that's a very good question. I would say that it wouldn't be too expensive because even when we buy it just to do our stuff in our mice, it's actually quite a cheap drug. It wouldn't be a therapy that would come up something like Luxturna where you're paying, hundreds of thousands of dollars for it. It would probably be a relatively cheap one.
Well, that's almost the holy grail, isn't it? If you've got something that works for all the conditions, then it doesn't have to be as specific as something that, say, Rick is working on. It gives a protection against all the conditions.
Definitely the holy grail.
Yeah. Okay, well, I don't know that there's anybody else. If no one else has a question, I think we might close there. So I just want to say thank you both very much for your time today. And it's just so fantastic to hear about these exciting new developments and think about where we're heading in the future. Thank you to you and thank you to the participants for coming. And I hope you found it was informative. If you still have more questions, please don't hesitate to contact us and we can try and contact our presenters and put you in touch with them. Now, when you leave the webinar, you'll be redirected to a very short survey just so that you can give us some feedback on this session, and it gives us really important information about planning for the future. So we've been recording this session and we'll send out a copy of that recording or a link to that recording in the next couple of days. And we'll also put it on our YouTube channel and on our website. Thank you all very much for coming. That's the end of the webinar, and enjoy the rest of your day.
Thank you.